So next, let's think about how to analyze the data. Right? In the typical analysis for the photochemical reaction, we use the kinetic analysis. We use the steady state approximation, which indicate the intermediate species concentration doesn't change during the light radiation. So intermediate species meaning that, for example, like excited state. And then here the things, at first we think about several kinetic processes. For example, initially the molecule is excited to the singlet excited state, and from the singlet excited state, so due to internal conversion, it's decayed, and the fluorescence emission is decayed, and also intersystem crossing is changed into the triplet excited state, like this. So we just make kinetic analysis for these processes, and then study the quantum efficiency, which is measurable in the experiment. So at first, for example, let's see when we add the quencher, meaning the fluorescence quencher, right? So meaning that by addition of this reaction, so A1 excited state is quenched by quencher, and the rate constant is given with KQ1. And then this 1A, the first singlet excited state, is intermediate species. That's why during the light radiation, this concentration doesn't change during the reaction. That's why this change can be regarded as zero. And how can you write this equation? The formation of A1 star is proportional to the light intensity. And this one decayed due to the internal conversion and fluorescence emission and intersystem crossing. And also, now you add the quencher, so KQ1 times Q is proportional to A1 star. So this is a differential equation of singlet excited state. Okay? And applying the steady state approximation, as I mentioned here, it can be zero. And also at first, let's think about when no quencher was added. I will tell you the reason later, soon. So by take this equation equals zero, this singlet excited state concentration can be written as KD1 plus KF plus KIST over I, right? And then the quantum efficiency is something like this. So I is absorbed light photon, right? And then the fluorescence rate is fluorescence rate constant times singlet excited state concentration, right? So this one is substituted here, and you could obtain the quantum efficiency of fluorescence like KD1 plus KF plus KISC divided by KF, like this. And similarly, if you assume that when the Q equal not 0, in this case, A1 star is KD1 plus KF plus KISC plus KQ1 Q over I. And the similar process you can obtain that you can obtain that the phi f is equal to KD1 KISC plus and KQ1 Q over K. Yeah. It seems a little bit complicated, but by doing this, by using this equation, this and this could be measurable. Without Q, without quencher, you can obtain the quantum efficiency, and also by addition of the quencher, you measure the quantum efficiency. And then you take the division by phi of f0 and phi f, and this could be much simplified by, you could just calculate this, and then KD1 plus KF plus KISC over KQ1 Q. So this is the experimentally observable, and the left equation is the function of the experimental parameter of Q, right? So by changing Q and obtain the experiment, you can make a comparison, right? And looking at the data here, so this is a whole process of the S1 singlet excited state decay, right? So the internal conversion and fluorescence emission and intersystem crossing. So you can make this one as a lifetime of the S1 singlet state. And then it can be much simplified as 1 plus KQ1 tau S Q, right? So now it's getting easier to understand, because you take this one in the vertical axis, and by changing the Q, and you can make a plot. And this equation indicate this has the slope of KQ1 tau S, right? So you can know the lifetime or the quencher rate. And also this one becomes the linear line. It means indicate that the assumption you made for the reaction is correct. But if this assumption is not correct. For example, in this equation, we assume that the singlet excited state reacted with quenching, but it might not be true because the triplet state might be reacting or some other reaction processes are involved. It's going to become even if you make a plot, it's not become the linear line. It indicates you have to rethink about the chemical or photochemical reaction processes, right? So this is how to obtain the reaction. So here's another type of another different reaction processes. So which is dimerization of benzophenone, which was introduced in as an example of photochemical reaction, including the initial process and final processes. Let me rewrite this process is different version a little bit actually. So we write this one BP, and this one BH is reacting with quite similar but different. Right? And in this reaction, the limiting process of this reaction is actually like this one is initially excited to the triplet state and reacting with BH and becoming generating the two radical species by reduction. And we define this KR reaction. And finally, it's coming to this. So since we know that this one is limiting reaction process, and this one just write BP. Okay, so we can use the no, that's not, this one is not written BP. So using the steady state approximation, since this BP star, so triplet excited state can be regarded as the intermediate species, we can write we can make the differential equation as formation of BP3 is equal to I and minus this is since triplet state decay due to the internal combustion given by the KD3 and phosphorescence and reaction, the above reaction KR BH, right? And this one is proportional to this one is proportional to the BP3 obtained as sorry, this is missing, and then BP3. And this could be written like this. So you could obtain the BP3 is the I over KD3 plus KP plus KR pH. So similarly as before, we can define the phosphorescence. In this case, the quantum efficiency, which is given by like this, and then it can be given by K. If you calculate this, Kp plus KRBH, and we think about when we add the quencher reaction too, and this is Kr and ISG. Actually, it's a bit complicated, but you need to have a ratio for intersystem crossing to generate the triplet state, like this. And it's getting more and more complicated, but just take the inverse of this, and you will obtain like YISG over 1 plus K YISG tau triplet BH, and these calculations are included in the after lecture exercise. So please try for yourself to do this calculation. But I will just show the answer for this. And KQ3Q. So it's now controllable by the BH concentration and the Q concentration to node. So the key point is we have to assume that first what kind of reaction is happening, and then taking express the measurable quantum efficiency with the controllable chemical concentration, and then in many cases just taking the inverse of this and make the calculation can be judged by from the experimental result. For example, in this case, the quencher concentration is constant, and then these values must be inversely proportional to BH concentration. So if this one is if you take the BH concentration, BH inverse concentration, and 1 over benzophenone quantum efficiency, and if this one is a linear relation, you can obtain your assumption is correct. So like this, the thing is you have to make steady state approximation by using the kinetic analysis, and then compare the experimentally observable or amount with some control concentration. Okay.